Hello friends, welcome to Biomass Learning. This video is regarding production of penicillin. Industrial production of penicillin can be broadly classified into two processes, namely upstream processing, referring to the processes before input to the fermenter, and encompasses any technology that leads to the synthesis of a product. It includes the exploration, development, and production. Downstream processing, referring to processes done to purify the output of the fermenter until it reaches to the desired product, such as extraction and purification of a product from fermentation. Medium for penicillin. The carbon source for Penicillin chrysogenum usually comes from corn steep liquor and glucose. Medium also consists of salts such as magnesium sulfate, K3PO4, and sodium nitrates. These salts provide the essential ions required for the fungus metabolic activity. Heat sterilization. Medium is sterilized at high heat and high pressure. The pressurized steam is used and the medium is heated to 121 degrees centigrade at 30 psi pressure. Fermentation. It is done in a fed batch mode, as glucose must not be added in high amounts at the beginning of growth. The fermentation conditions for the penicillium mold usually requires temperatures at 20 to 24 degrees centigrade, while pH conditions are kept at 6.5. Seed culture. The seed culture is developed first in the lab by the addition of Penicillium chrysogenum spores into a liquid medium. When it has grown to the acceptable amount, it is inoculated into the fermenter. The medium is constantly aerated and agitated. Carbon and nitrogen are added sparingly alongside precursor molecules for penicillin fed batch style. After about 40 hours, penicillin begins to be secreted by the fungus. After about 7 days, growth is completed. The pH rises to 8.0 or above, and penicillin production ceases. Removal of biomass. Filtration is carried out, as bioseparation is required to remove the biomass from the culture. A rotary vacuum filter is commonly employed for filtration. After filtration, phosphoric acid, a non-oxidizing agent, is introduced to decrease pH from 8 to 6.5 so as to prevent loss of activity of penicillin. Addition of solvent. Organic solvents such as amyl acetate, butyl acetate are added to dissolve the penicillin present in the filtrate. At this point, penicillin is present in the solution and any other solids will be considered as waste. Centrifugal extraction. Centrifugation is done to separate the solid waste from the liquid component which contains the penicillin. The supernatant will then be transferred further in the downstream processes to continue with extraction. Extraction. The penicillin sodium salt is obtained from the liquid material by basket centrifugation, in which solids are easily removed. Fluid bed drying. Drying is necessary to remove any remaining moisture present in the powdered penicillin salt. This was all. Thanks for watching.